So we are going to see about siderophores. So siderophores are small ion binding molecules. So usually these siderophores will be secreted by the microorganisms. So what type of microorganisms are capable of producing these siderophores are bacteria and fungi. So what is the main function of this siderophores? So siderophores usually it is used for promoting the growth of the microorganisms and also the growth of the plants. Okay, so these are the different functions of siderophores, and in general you call these ion binding molecules as ion chelators. That is, the main function is to transport the unavailable source of iron into an available source of iron. And you have different types of siderophores. So mainly when you focus on the types, so one major class is belonging to the microorganisms. So the example is bacterial siderophores, and the other one is siderophores that are produced by the plants, which are called as phytosiderophores. And next we move on to the structure of the siderophores. So when you take the structure, so structure mainly it will be focusing on the chemical structure of the siderophores. So when you examine the structure, so the structure will be a hexadentate complex or it will be an octahedral complex. And if in case there are two different molecules joined together, the ion will be in the center part, and then the two side chains will be belonging to the siderophores. So you call these structures as bidental structures. Okay, and then what is the main aspect of the siderophore is reduction of the ion molecules. And next we move on to the major groups of siderophores. So the major groups of siderophores are catecholates, hydroxamates and carboxylates. So examples of catecholates will be phenolates, and then carboxylates mainly those are derivatives of citric acid. And how these siderophores are transported. So whenever a nutrient is given for the microorganism or for the plant, it has to be transported inside the cell. So for the transportation into the cell, you have different types of transport systems. Okay, so you could have learnt about symport, antiport. So all those transport systems are required for transportation of any particular nutrient source in and out of the cell. So first we will be focusing on the hydroxamate siderophores. So hydroxamate siderophores usually they are called as ferrichrome. So ferrichrome, it is produced by Eusteligo, and fusarinum, it is produced by Fusarium, and ornibactin, it is produced by Burkholderia. And next we move on to different kinds of siderophore producing organisms. So usually E. coli. E. coli is a common example for any particular genetically modified experiments. So this E. coli is also used in siderophore production. That is, the name of the siderophore produced by E. coli is Enterobacter, and Bacillus subtilis, it is producing Bacillibacter, and Vibrio cholerae, it is producing Vibriobacter. So next we move into the different types of ligands. So I told you ligands are a combination of molecules. Okay, so there are some kind of mixed ligands are also present in the siderophores. So Acetobacter, it is producing acetobactin. Pseudomonas, it is producing pyoveridin, and Yersinia, it is producing Yersinia bactin. So all these are pigmented siderophores. So visually we will be able to see the production of siderophores when we are growing the microorganism under laboratory condition in a defined medium. We will be able to look for the visible production of pigmented siderophores. Okay, and next we move on to the genes that are responsible for siderophore production. So how these siderophores are produced by the genes that are present in the microorganism. So whenever there is any particular gene expression, so we have different types of genes that are involved in production of siderophores also. Okay, so you have a origin of replication gene, then you have a transcription termination control, and then you have a termination sequence. So like that, you also have an inhibitor, initiation factor, and also repressor. So all these genes should be available for any kind of product production. So when you take repressors and inhibitors, so what is the main activity of this repressor is to reduce the level of siderophores. So these repressors has to be used for siderophore production. So we will be looking for two different types. So one is under higher iron concentration and the other condition is low concentration of iron. So under higher concentration of iron, what happens is the repressors will be activated. So what is the work of repressor is to stop the iron production. So when there is abundant amount of iron produced, the repressors will be coming into action. It will stop producing the siderophore. Okay, and then in the lower concentration of the iron molecules, what happens is the repressors will be activated. Okay, and then they will be going for the production of siderophores. So this the main action of siderophores has to be kept in mind, and it is the same pathway as how it is going in different kinds of microorganisms. So whenever there is an initiation factor and a promotion control sequence, so it will initiate the transcription of the genes. So whenever the iron has to be produced, this particular process will be initiated, and after the iron is produced, it will go for inhibiting the production of iron. So that is the concept behind the siderophore production. So next we move on to the mechanisms that is normally occurring in the bacteria. So when you take bacteria, you have AT rich gram positive and gram negative bacteria. So both these AT rich bacteria and GC rich bacteria, so GC content and adenosine thymine content. So these two types of bacteria are noted, and then they have compared the production of siderophore production in these kind of bacteria. So when you take AT rich bacteria, what happens is the production of siderophores, it is regulated by a repressor protein. So the name of the repressor protein is ferric uptake regulatory repressor, FUR. And then you take another kind of bacteria, that is GC rich gram positive bacteria. So the example is actinobacteria. So here you have a repressor that is named as DTXR. So DTXR, the expansion is diphtheria toxin repressor. So these two repressors has to be in action when the organism is going for siderophore production. So all these siderophores are produced by the microorganisms. So after production, how it is moving into the cell. So how it is moving is the siderophores which are produced, it will be moving into the cells with the help of certain receptor molecules which are present on the outer membrane of the cell. So these receptor molecules has to identify the siderophore, and then the siderophore will go and bind to the receptor and it will be transported inside the cell. So one example is given, that is in case of fungi and eukaryotes. What happens is the iron molecules will be reduced. So Fe3 plus will be reduced to Fe2 plus. So these receptors are helpful in transferring the siderophores, and then it is moving into the cytoplasm. So this particular mechanism is common for all types of siderophores that are produced by microorganisms and also by the plants. So the iron molecules will be reduced. So Fe3 plus is getting converted into Fe2 plus. So what happens is unavailable form of iron is getting converted into available form of iron. And next we move on to the siderophores that are produced in the plants. So the siderophores that are produced in the plants are called as phytosiderophores. The phytosiderophores, the expansion is the siderophores that are produced in the plants. So in an iron deficient soil, so that is in a laboratory experiment, what they will do is they will take the soil that is having a lesser content of iron, and then they will look for the production of siderophores. So what happens is the iron binding molecules will be produced, that is the siderophores will be produced, and then it will be secreted into the rhizosphere region of the root, that is the region that is near to the root, you call it as rhizosphere region soil. So in that particular area you can see the production of siderophores. So how it is transported into the plant is through the mechanism, that is proton symport mechanism, and then it will be transferred into the phloem of the plants. Okay, and how it is carried is, it is carried by nicotinamide. So this particular component, it will be acting as a carrier for transporting the iron molecules. So here also the iron, unavailable form of iron will be getting converted into the available form of iron. And next we move on to the common type of microorganisms that are producing siderophores. So the name of the siderophore is ferroxyamines. So that is produced by Bacillus, Arthrobacter and Nocardia. So all these siderophores, the main function of the siderophores is for the growth promotion of plants, and also in turn it increases the growth of microorganisms that are normally present in that particular soil. So you have some indigenous microorganisms that are normally present in the soil. So these type of organisms also will be increased. So when this organism growth increases, it also promotes the growth of the plant. So mutually it is benefiting both the plant and also the microorganisms. So these are the common aspects of siderophores. So what all you have to concentrate is, first of all you have to define what is a siderophore. So you have to give this particular terminology, that is iron chelating compounds that is produced by the bacteria, fungi and also plants, and how it is transported into the cell is through the cell membranes. And then you concentrate on the functions of siderophores, then types of siderophores, and what are the major groups of siderophores, that is catecholates, hydroxymates and carboxylates. And then you give examples of each of these organisms that are producing siderophores, and then you have to explain about the genes that are coding for siderophore production, and what is the condition that is happening in gram-positive bacteria, that is AT-rich gram-positive bacteria and GC-rich gram-positive bacteria. And then finally you say about the applications of siderophore. So how it is used for promoting the plant growth.